Hello friends, in previous lecture we shall discuss about the biological process or some biological tools. Now these biological tools discuss the first enzymes, some vectors or cloning vectors are very important tools for these biological processes. Now these enzymes there are so many enzymes are utilizing for this process. One of the important enzyme like the restriction endonuclease. This enzyme is a very important for the specific cut of the DNA fragments. And other enzymes is also important for this process. And now the next one tools is vectors. And this vector is known as a vehicle. They can transfer the genetic material from one cell to the another cell, and it is called the vector. Now some vectors like the cosmid, plasmids, lambda-phage, bacteriophage, these are the different vectors are used for the genetic engineering or transfer the genetic material from one cell to the another cell. Now this next one points methodology of RDNA technology. Now in this lecture study this methodology of RDNA technology. Now this RDNA technology means the recombinant DNA technology. In this recombinant DNA technology, the several steps are involved for the recombination DNA technology. There are some steps or RDNA technology or gene cloning methods. There are these steps. First one is the isolation. Now this first step of the RDNA technology, it is the isolation of the DNA from donor organism. Now this first step is the isolation, means this genetic material or DNA they can isolate it from the donor cell. This donor cell or the specific DNA or desired DNA to be cloned, and they can obtain from the other cell or the specific donor cell. This DNA is isolated. In this first step, now the second step, it is the insertion. This DNA is isolated and later this DNA is transferred to the another cell or a specific desired gene into the cloning vector. To this vector is important for this insertion process. Vector means it is a vehicle and they can transfer the genetic material through these vectors. And now the third step is a transfer of the RDNA into the suitable host or suitable component host. It is utilizing the transfer the genetic material into the suitable host in third step. And now the fourth step is selection of the desired gene or this transform host cell. And now the next one, multiplication and expression. These 5 steps are detailed study. Now the first point, it is isolation of the gene or genetic material. Now this genetic material, it is isolated from the donor cell. Now this one of the cell, the specific donor cell, this DNA can isolated. Donor cells means this one of the genetic material or DNA is isolated from these cells. This DNA, it is a coiling structure, and this DNA, it is firstly isolated. Now this isolation of the desired gene or desired segment of the DNA, and particularly this DNA is a thread-like structure or it is a double-stranded helical structure and specific cut of the DNA. This one of the point, this target point, it is a target gene or DNA for selection. Firstly, isolation of this genetic material. Now this desired gene, they can isolate various methods. Now this gene is isolation or some genetic material is isolated from the donor cell, other cells, that time some steps like the removing this isolated material, they can remove from the other cells and they can purify the several techniques. Now these techniques are using the several techniques for the isolation of the desired fragment of the DNA. Now this segment they can purify the DNA at for the using the cleavage is using the restriction enzymes are also utilizing for the isolation of the target DNA particularly the restriction endonuclease. This enzyme is used, restriction endonuclease. This restriction endonuclease enzymes is very important for the cut the specific cut, means this enzyme most important function for cut the specific fragment of the DNA, and this specific point means the target point of this host cell. This DNA they can isolate or they can present in the cleave and the specific target point. This restriction endonuclease is useful for the cut the specific DNA fragments, and now these enzymes they are also cleaved to specific site called the restriction site. Now the restriction site means the some nucleotides or nitrogen bases are found in these DNA strands, and these some sequences are found in these strands, and these enzymes, specific enzymes are utilizing for the restriction site for cut these enzymes. Now these several types of the restriction endonuclease are using for the cut the fragments of the DNA, but this DNA having the cleaved segmented structure because present of the number of nitrogen bases in this strand, and now these two strands they can specific cut is takes place. Now this specific cut like these sequences of the DNA are found, and cleaved DNA like these fragments is cut the desired gene. They can also present the sticky and stagger cut. Means this DNA is cut into two sides, also the straight cut and it is the opposite direction or the stagger cut, and these they can having the sticky ends, and these two cuts means the desired gene they can isolated or desired DNA is isolated in these steps, and now this they can select for the cloning. This first donor cell they can isolate the specific DNA or this DNA is a origin DNA or it is also called the passenger DNA. Now this DNA it is a desired DNA they can transfer from the other cell, and this gene or DNA they can also save this DNA or they can obtain directly through the genomic library or cDNA's library. Means this isolation is takes place through these libraries, means they preserve the genetic material in these libraries, the DNA they can isolate it. Now the first point, it is the isolation of the DNA from donor cell. This is the donor cell, and this DNA they can isolate it through the specific enzymes are used, the restriction endonuclease, they can cut the specific fragment of the DNA. Now this is the first step of this isolation. Now this next step, it is the insertion of desired gene into the cloning vector. The desired gene, the specific gene, they can clone through the vector. Means this they can insert the gene through the vectors. Now this vector, it is important for the transfer the genetic material. Now the vector means vehicle. There are some vectors like cosmid, plasmids, bacteriophages. These are the some vectors, and through this vector, this genetic material they can transfer to the this isolated DNA is transferred through this target cell. Now this first one, the vector is used. The vector, second step, using the vector, and this vector like a plasmid or bacteriophage. These are the vehicles or vectors. Now these vectors, this is the structure of the vector. It is a rounded structure. It is the structure of the vector. And this vector, it is cut the specific point. This vector are cut. Now this vector, this is a vector DNA. This vector is cut for the specific point, and now this DNA, it is inserted the small fragments is cut for the DNA molecules, and now these DNA molecules or the fragments, they are cut through the enzymatic process, and this fragmentation by the restriction and the enzymes. Now these fragments, this fragmentation process, several cut of the DNA and select the specific cut of the DNA, and they can join to these vectors. Now these firstly, second step are involved for the insertion of desired gene into the cloning vector. This cloning vector is important for the transfer the genetic material through this vector. Means the desired origin gene or the DNA, they can introduce the cloning vector or the vehicle. Now the origin DNA, like the means this passenger DNA, they can inserted into the cloning vector through the these commonly using these vectors like the plasmid and bacteriophage. Now the next stage, third stage of this rDNA technology is the transfer of rDNA, recombination DNA. Means this recombinant DNA, they can transfer to the suitable host or cloning organisms. Means this one of the these vector and joining this DNA, join to this DNA into this vector. This joining of the target DNA and cloning vector, joining target DNA to cloning vector. Means these DNA are joined to these vectors. Now this vector is a vehicle or transfer the genetic material through the target or this another cell or host organisms. Or the third step, means this target DNA, this is the target DNA. Now the next step, this they can introduce this vector into the transfer into the suitable component host. Now this host cell, this host cell, and this insertion of this target DNA through this cell, this one of the bacterial cell is host bacterium. This is the bacterial cell, host cell, and this bacterial cell are used for the this DNA is transferred through this bacterial cell. They can introduce the bacterial host cell, introduce to bacterial host cell. This bacterial host cell means this genetic material they can transfer we insert the genetic material through this bacterial cell. This bacterial cell is one genetic material, and with the help of this vector they can insert into this bacterial cell the genetic material. Now this transfer the DNA through vector to target or the bacterial cell. Now this transfer the host cell containing the DNA molecules, and later the identification and amplification of the DNA. This next process next step of the rDNA technology, recombination DNA technology, is the transfer of the rDNA or through this rDNA technology into this DNA molecule through vector or vehicle through the bacterial cell, and later the selection of the transform host cell, and they can multiply. This multiplication means this host bacterial cell introduce this target DNA, and later this host cell they can multiply the genetic material or multiplication of transform the host cell. Therefore the multiplication or formation of the these double copies or these target DNA molecules, or the cell is a multiplication and this lastly the multiplication takes place, and these proteins they are encoded by insertion of the DNA. Now this cell multiplication or protein products form in the clone DNA. Now in this bacterial cell form the clones, or many target DNAs are produced, and they can forming the protein encoded inserted DNA. Now this target DNA they can multiply it, and these proteins code means genetic materials are formed. In this rDNA technology, there are several steps are involved for this recombination DNA technology. The first one, isolation of the desired gene. Next one, insertion. This insertion of the specific DNA fragments to the target DNA to the host cell. And now third step, it is a transfer of the rDNA to the suitable host, means bacterial cell or these bacterial organisms, like cloning takes place in also plant biotechnology. This E. coli bacterium are also using the agrobacterium tumefaciens. Now these are the component host cells are utilizing for the transfer genetic material, or transferred cell are used, and this origin DNA also transfer directly into these naked cells or protoplasm of the component host cell. Now the next step is a selection. Now this selection of this material of the host cell, and lastly the selected material, like means vector is most important, like the different types of the example of vectors, like the PBR, the means plasmid, this P322, this also plasmid as a vector, and they can contain the different marker genes are having this transform host cell. And now the last step is a multiplication, and this multiplication is this host cell they can multiply this genetic material or DNA, means recombination DNA technology. This cell multiplication takes place on this cell, they are separated through screening process. Now this step they are transfer the cell and they can introduce in the culture. Now the last step, in these steps the expression of the gene to the obtain desired products. Now these next steps, they can involve the production of the desired gene. It is also utilizing the they like alcohol, enzyme, antibiotics, and finally these desired products, it is separated through the purification process, and through these are the using the suitable bioreactor. Now these are the 5 steps involved in the rDNA technology, recombination DNA technology. Firstly, this DNA, they can isolated the donor cells with the help of vector. This vector is a vehicle for the transfer through this vector transfer the genetic material from this one of the cell, bacterium cell, host cell, and this host cell, this introduce the bacterial cell, this genetic material, this target DNA is introduced into this host cell. Later they can multiply and number of the copies are produces. Now this in this lecture we shall discuss about the rDNA technology steps of this. There are 5 different steps are involved in the rDNA technology.